All right. Hi, everybody. This is a supplementary lecture for 1121. It'll probably come in more than one part. I would like to finish up as much of the central nervous system as I can. And this is an area, and I'm trying to do this by looking at the exam. I'll get this open for you. And this is where I left it. I mean, I got a little farther in the 9 o'clock class, but I want to go over this again without going crazy about what it actually does. So we had just completed, if you recall, I'll back up a little bit just so you can see it. We had already basically finished the epithalamus, the diencephalon, all of those things. And I think that was covered thoroughly for everybody. Again, now that we go farther down, and you can see it here, now we're into the brainstem. So this opening and passageway is called the cerebral aqueduct, and it opens into this relatively large chamber called the fourth ventricle. And you can see that little reddish guy there. That's one of these choroid plexes. And they have them in all of the ventricles. There's one for the third ventricle. And effectively, that's where the cerebrospinal fluid is effectively filtered and finalized and produced. The ependymal cells play an important role. It's a good illustration to see that. And I'm sure we're going to see the back. This is a famous one. This particular image is called typically called a hemi or half section, and it really shows a lot of the anatomy, I think. So the fourth ventricle again is very significant because it has those areas of outflow where the spinal fluid, when we get to this later, and this might be in this presentation, certainly will be one of the ones that I send to you today or in the very near future. I'm doing this Friday morning on the 21st. And so we'll look here. So the cerebral aqueduct gives way to the fourth ventricle. And then as we get to effectively, just as we get into the medulla here and all the way down, it gets very narrow. It's called the central canal. And effectively, that is how should I say it? That's really a very interesting area as well. I never know where to put anything anymore. So I'll move on and then see what I can come up with here. So I'll probably do this first one in a mini shift as I have to go look for something. Nevertheless, so that this is, beg your pardon, I don't have a cursor. I don't have a mouse, so I have to do something else. This is the midbrain, this is the pons, and this is the medulla oblongata. These are the areas. So the midbrain is separated. The back portion, known as the corpora quadrigemini, and they're called the inferior and superior colliculi, are really important bits of gray matter that help to basically identify quick reactions to sight and sound, like you're a siren. You turn your head. So they're really important. They control reflex action of the head to both light and sound. And the anterior portion here, if you want to call it the midbrain proper, and we'll see the section, is other than cranial nerves that their nuclei are there, the cluster that make those cranial nerves, where they're basically is about neurotransmitter production, which I've commented on before. You'll have the red nucleus and you'll have the substantia nigra, and you'll see those very, very distinctly. After that, as we get to the pons and the medulla, it's all just cranial nerve roots and a lot of nuclei that play a really significant role in regard to this is all about, the brainstem is all about stuff we don't control. Autonomic or automatic functions, breathing, circulation, heart rate, all of those things, blood pressure, just name it. All comes from these areas that are there. So remember the choroid plexes, they're important. Remember the ependymal cells, that's part of it as well. And just shows you again. And this is now you're looking at it. I mean, they've made it neat colors. Pons very distinctive because the gyri and sulci are somewhat transverse. This is not certainly, but you can see all the cranial nerves that emanate here that are part of it. So it's a busy area for that, but unfortunately damage here doesn't work particularly well. Big problems that are there. And here you can see it again. If that interesting sort of neat thing is the optic tract. There's a pituitary hanging off that stalk of the infundibulum. You can see the colliculi in the back. And again, the largest by dimension, by girth of all, it's three nerves, basically called the trigeminal. That's what means it has three ganglia that are associated on the pons that come out, very important. And then the cranial nerve is really at the junction here between the pons and medulla, just a bunch. I mean, if we were doing cranial nerves and we were doing peripheral nervous system, that would be a big issue that I was going over anatomically. Here it is from the back. There's that choroid plexus, etc., etc. So the illustrations are good. So the midbrain, pons, medulla, good point to make here. Basically, like the spinal cord, contains nuclei embedded in white matter. That's about it. Okay, autonomic or automatic behavior, lots of tracks going up and down. All but two of the cranial nerves, that's olfactory and optic. They're the only ones that go optic to the thalamus, olfactory directly into the insula, right into the cerebral area. I'm sure you saw that illustration or something like that when you talked about cranial nerves with Dr. Robbins. No big deal. I don't want to go crazy with nomenclature about peduncles and stuff like that, but really that's the name for those important pyramidal motor tracks. I'll show you where the pyramids are. Those are the famous corticospinal tracts. And I mean, and all this other stuff, I don't have to worry about periaqueductal gray matter, et cetera, et cetera. Again, these are visual and auditory reflexes. They're called the colliculi. This name it means four ganglion bodies, if you want. And then this is a big player. Don't forget this. It's linked to Parkinson's. It goes from the substantia nigra to the corpus striatum. It's about dopamine production. You must remember that. And the red nucleus is interesting because the reticular is sort of your way you wake. So it goes all the way through your spinal cord, your brainstem, all the way up into the brain. This series of passages and nuclei, basically cell bodies and passage and axons that carry this message. It's like when an alarm clock waking you up, somebody shaking you, waking you up, somebody turning the lights on, maybe you don't have the shades drawn and you're sleeping, and all of a sudden the light comes in and hits you, bang. That's your reticular formation. That arouses, and not arouses in a sexual sense, but arouses and awakens, that type of a sense. So that's interesting. This is the cutaway or the transverse. See the red nuclei. You can see the substantia nigra. These guys are those immense. These are going to become the pyramids when you see the medulla. These are all projection fibers going up and down, etc., etc. The pons is just it's a relatively large area compared to the size of the other two, and that's about what you can say with it. Again, up and down fibers going back and forth. It's really the relay center. The cerebellum, which we're going to get to, sits behind it almost completely. Okay, but a little bit to the midbrain side. There's a little bit of the cerebellum to the sort of back from the midbrain or posterior to it, as well as from the medulla oblongata. But you got up and down fibers, and you have fibers front to back kind of fibers. If you want to think of it this way, the giant trigeminal nerve. Again, so and the reticular formation for awake, breathing, everything. It's almost like, and you'll get to it in AP2 physiology. It's almost like setting the beat for your heart, setting the beat for your respiration, making sure that your blood pressure is adjusted. There's a constant relay of fibers from the pons and the medulla conducting business in that regard, as you will see. Again, lots and lots of fibers are in this area, scattered with nuclei. Here's the giant trigeminal that are there. Here's some of that reticular formation for arousal. And you can see the relatively large amount of the fourth ventricle. It's a very interesting area. We're not going to get into all the other things that are associated with it, though. Again, in a more advanced anatomy or the graduate anatomy, certainly would. And if you don't get confused again by these terms, peduncles are just white matter tracks. White matter tracks. They just get, I don't know why, they were sort of like, I guess they represented columns a la, remember, so a lot of it's generated from Latin. So a peduncle was sort of one of those big columns that's holding up buildings. And that's kind of where they viewed these. And then lastly, the medulla. This is the most inferior aspect. This is damage there is really, all of the control centers just go. And at that point, people have a very, very limited chance of survival. Tumors there, trauma there, very significant. So, and effectively at the foramen magnum, it blends is a good word. It's not like you go from one to the other. There's a relatively quick but still gradual transition from the anatomy of the medulla to the very distinctive anatomy of the spinal cord. Okay, and again, the choroid plexus, big player with regard to forming is an okay term, but again, it's to me it's almost finalizing it would be better because it's really an extract of plasma and it's adjusted. But that's just me. Again, you're going to see these mounds in the front, which are the pyramids. They get their name from that. And a really important word to remember is called decussation. Decussation, it just means crossing over, but it's a fancy name. And again, it's one of those things that one of those names that you got to remember. One, which is going to be on the test. Two, that's the kind, because you can't just say, oh, the pyramids cross over. They decussate. When fibers go from one side to the other side, that whole idea of contralateral control, right side of your brain telling your left hand what to do, et cetera, that's because the fibers decussate, and they decussate or cross over in the pyramid. I know it would be easier to say crossover, but what fun would that be? And not worrying about cuneatus and gracilis or the different cranial nerves. And there's that same illustration again. And the same illustration. Here's what it looks like. You can see the choroid plexus is very distinctive the way they've illustrated it here. Again, the olivary nuclei have different roles, and we don't have to get into it. But these are those pyramids. So they had that base kind of wide base, kind of narrower apex. I don't know if it's such a great idea of a pyramid, but we'll take it for what it is. But lots and lots of cranial nerve nuclei are all over here. So there's so many controls. Damage there, which is critical. So reflex center overlaps with the hypothalamus, heart, blood vessel, blood pressure. So you can imagine what would happen. Respiratory. And again, interestingly, it's sort of there are links between that and the pons when you get to physiology that really control things like rate and depth of breathing. So it's not just isolated that one area. And if you didn't know it, these are all protective reflexes. This is the way we eject toxins before they become a problem. This is something that's irritating our diaphragm, and we can expel something. We usually suggest or something. I mean, it obviously has other causes. All of these types of things, irritation, these are foreign particles, etc. And these are things that you have to be able to do in order to survive, by and large. The second largest and an extraordinarily important area has the vast majority, a greater amount of synapses, nerve connections, because it's constantly coordinating information about movement. And it sends that information, it collects it from the extremities, it collects it from other parts of the body, the head, the neck, the torso, the brain itself. It funnels information up and down and directly across by way of its peduncles or white matter tracts. It's located immediately behind the pons in the medulla, not so much the midbrain, maybe just a teeny tiny bit, and inputs from everywhere. Inputs from the cortical area, the brainstem, all those receptors, all those proprioceptors, all the information so you know exactly where your body is, what its position is, what it's up against, what it's feeling. Is it hot? Is it cold? All those things that we process without even thinking about it. And very, very large. It's got two mounds. And again, the sulci and gyri are very distinctive because they're much more horizontal than vertical if you look up at the cerebral cortex, up at the superior portion of the brain. So they're transversely oriented. They call them folia, don't care about the lobes, care about this. This is called the arbor vitae. That's the white matter, and it really defines it. That white matter tract is very because arbor vitae means tree of life, and tree of life is basically what anatomists looked at and said, "Gosh, this looks like the religious tree of life that is." I'm not going to go through the anecdote again, but it was the name of the synagogue where that terrible tragedy occurred in Pittsburgh, where a guy graduated from high school was murdered. We'll leave it at that. So the arbor vitae is they felt that this was really an essential part of life, and they named it arbor for tree, vitae for life. And it's a white matter, very distinctive. Purkinje is a name that you'll see it when you get to the heart as well. And this is the communication passageway. You don't have to know that. And they even have a homunculus for the cerebellum as well. But this is what it looks like. So this is that very, very famous. Here's the fourth ventricle. It's one little, and it's a large ventricle compared to the aqueduct here, and certainly the central canal down there. And you can see the distinctive way that this is transverse. And this is white matter. And there's a lot of gray matter on the exterior. So this is the other area where we see a cortex. So we have a cerebellar cortex and that very interesting white matter. Here they are giving it to you more schematically, but still the idea is the same that are there. And again, these passageways that go forward and backward are called peduncles. You don't have to know that. This is what it looked like. And so they even have a homunculus drawn for this, as you will see. I'm not going into it, but the big deal is all fibers are ipsilateral, same side of the body. You're getting the information, distributing the information. If it's coming in on the right, it's being processed on the right, distributed to the right, etc. And obviously opposite for the left. All the neat things it does, we'll leave it with that. Impulses, proprioceptors, informing body position and movement, best way to move, blueprints, and then you get into things like motor, muscle memory, which is very complicated but very interesting, particularly in sports, and other stuff. When we say neuroimaging, it's like those things that we looked at that light up. It plays a role. So when there's damage in there, and that's what I was telling you about alcohol, which definitely impacts it, and other medications impact it. Not that alcohol could be a medication, but other substances impact it. And when I say substances, I don't mean something that's necessarily a drug or illicit or anything along those lines. Okay, so when people have when there's damage in that part, there's a lot that goes wrong with it, the way I speak. And this is like the homunculus. I mean, it's effectively trying to map out areas that it might have something to. And again, so if this is like from my perspective, this is right hand, this would be right hand, and this would be left, etc. There, and that tidied that up very nicely. So we're done with that. I'm going to X out of it. Come back over to here and take a quick, because D takes long. I'm not going to do that one. C is basically the meninges. There's a lot of functionality in C, and I really don't do it for this course. The limbic system. The reticular formation is that awake and alert system that what we call arousal, and you see it's rather extensive. It's spinal cord. It goes out into the extremities, so when you're touching those sensors, and it brings it up wherever it's coming from. But the limbic system is that emotional system. I do have to point that out. Okay, and it's on the interior. When you open that brain, and you opened it in half like you're opening a book, on the outside is one, but the interior, we call the medial aspect, the part that's right against the midline, is where that limbic system is. And it includes cerebral structures and circles the brainstem, parts of the diencephalon, the big ones, okay, the amygdaloid or amygdala body, angry or fearful facial expressions, assesses dangers, less severe response. This is associated with rage. The amygdala or amygdaloid body, either term is used, okay. The cingulate gyrus, I'll show you where it is. I'm not going to ask you a question, but it's part of it. This is the one. Cingulate means it's C-shaped, and you see it surrounds the corpus callosum. So this gyrus of the interior portion of the brain, going from the frontal all the way back through the occipital lobe and maybe even into the temporal bit, that's the cingulate gyrus. Not a big deal. The amygdala is here. These are all structures of gray matter. There's the amygdala. There's the very famous hippocampus. So you have the hippocampus. You can follow the lines here, all in this area. Here's the amygdala or amygdaloid body. Oops. And again, this is the olfactory bulb. And again, smell plays a significant role. And you can look through this. Gives you an idea. There's a lot of names that are associated. The ones you have to know about. And here's that septum pellucidum, that divide. And technically, that's part of it as well. That's why I had one student whose son was born without that. And he's basically as normal as you and I, but he just had emotional issues. So it's interesting that there it is. So here you have the hippocampus, short-term memory, Alzheimer's, the amygdala or amygdaloid body, rage, sense of smell playing an important role. All factors you should remember. I don't know enough about psychosomatic illnesses to talk about it. It interacts, as you can see, with the prefrontal. That's the beauty of that prefrontal area and why it was so terrible when they began to lobotomize people. That's the term that they used. It's what we react. This is the world is through us with emotions. Whether it's like me when the Steelers win a game and I'm all excited, or when they lose a game, but this our interactions, things like that. When your kid does something well, or you do something well, and you see the glow in your parents' eyes, that type of stuff. It's all really important. And then particularly the hippocampus, unless the amygdala, for memory. And then I don't go into any of these eternally. I just need you to know it all over the place. Hypothalamus, thalamus, govern it's brain arousal, not other arousal. And it's called the reticular activating system. It has lots to do with sleep, injury, coma. A lot of this chapter deals with electroencephalography, and I'll buzz through it. I'm not going to ask you about it. I don't know enough about it to be able to teach it. It has a lot to do with sleep disorders. It has a lot to do with seizure disorders, which I'll comment on, but I don't plan on asking anything. And again, this also, they play a certain role. So think of it, when you're sleeping, your blood pressure changes, your heart changes, your breathing changes. So it plays around. And then when you awaken, it changes. That's where that happens. And it's a good illustration to show you. It can be here, it can be auditory, all over the place. So you can see all of these things, whether it's auditory impulses, pain, touch, light, all those things can activate it. Let me just summarize here. The summary is useful. I don't do these because I don't understand them. Not that I don't understand them, maybe a better word. I don't understand. If I don't understand something well enough to convey it to you, what's important, then I'm remiss. In all of these things, I know a little about them. It's much more complicated. This would be, I would think the folks in psychology would have a better idea. That they're a little more familiar with it. We really don't have. We have a neuroscience class, but it's basically the same kind of material, just gets it in a little more detail. So again, this is the idea of Broca's and Wernicke's, and you can read through it, and you saw that illustration, that different kinds of memory. And it's interesting. Again, emotional memory works. Okay, motor memory, you know, all of these kinds of things. There are different ways. You don't have to any of those. Short term and long term. I tend to think that I don't know about you, but I'm fortunate enough to remember more of those things without having to go to the long term. But long term has no. And all this type. This is the mistake. Okay, repetition and practice. It may be good for learning how to do certain types of things. It may be useful in sport, but for studying for tests, just sitting there, and we call it rote memory. You just sit there. What part of the brain I have to know about the medulla? Medulla this, medulla, smallest area of the brain, medulla. You pound that in. It's useful to remember for the test. It's not useful to remember long term. So maybe for a lot of the stuff in this course, that's fine. Because you're not going to be asked no one is going to ask you if the dysfunction and the reason that they are in the hospital is because of an imperfection or irregularity in their medulla oblongata. Guarantee it. Just saying. And you can go through this stuff with memory again. I don't even try. Oh, I'm not going to go crazy about amnesia and things like that. They're all part of it. But one of the ways we use it, particularly for seizure disorders and sleep disorders, they still do, is it's called an electroencephalograph, or EEG, very famous. They put the electrodes on your scalp, or they wear a cap that has the electrodes embedded into it. It's non-invasive. It doesn't hurt. And there's many of them, and they get a picture of brain waves. And we'll use it to look at sleep cycles and dream cycles. They'll be able to see what the changes in that when you have this thing called a seizure disorder, which is called epilepsy or an epileptic fit, because think of those are spontaneous discharges, short circuits, if you will. So all of these types of things. And it does help for other things like tumors and things, but our scanning technique is pretty good. Legally, this is about brain death. When we basically have flat EEGs, okay, over X amount of time, brain death is death. As far as I know, every state that is the legal definition of death. I don't want to get into that and advanced directives. For many of you in nursing, you're going to become very, very familiar with that, and it's extraordinarily important because a lot you must follow those advanced directives. And obviously, there's going to be certain exceptions, but if you know of an advanced directive, you must follow that advanced directive. And in case if someone does not want to be resuscitated, if there's a DNR, as we call it, I mean, it's different. I mean, I dealt with this with my mom when she had Alzheimer's, and I mean, and she was very specific. My brother-in-law is an attorney. He didn't draw it up because that would have been inappropriate, but his members in his firm drew it up, independent of him and my sister. And God bless them, they would never have done anything wrong. They were just wonderful with my mom, and effectively, we knew exactly what her wishes. I got a copy of it, very straightforward. If you feel there's any chance of success in resuscitating me and blah, 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 this, do so. But if, blank, and she set out the criteria, don't. I mean, like I said, she lived to be 93. And so typically they don't use the cap so much as they try to put it directly on the scalp. It's usually you need some type of a medium, like a lubricating agent, sort of like like a gel is the right word. And so it just allows the impulses to be conducted more efficiently. That's there. Very often it'll be for induced sleep, and because they want to see sleep EEGs, and this is a useful way of doing it for all of these things. And that there's different kinds of brain waves, but based on how they're measured, is what's called cycles per second. Don't know anything about it. Don't know anything about it. You can read through this. I've seen them interpreted. No way to do it. So basically, this is the idea of the short circuit or torrents of electrical discharges. And old days we call it grand mal and petit mal. Very bad, not so bad. But the reality is this now we call these tonic clonic, and this is the classic examples that you begin to see. You have to learn how to handle them. Don't be sticking things in their mouth. If it's before the seizure comes on and something can get in your mouth, that's fine. But once they start to seize, don't get your fingers anywhere near that because you want to be able to count to 10. Being very serious. And the most important thing, if you come upon somebody who's seizing, is to make sure, because if they're on the ground, that herky jerky can really hurt their head and do more damage, really, sometimes than the seizure. It's very stigmatizing because they lose control of bowels and urine, so they end up voiding and soiling themselves. It's not good. It is not associated with intellectual impairments. Be aware of that. Sometimes in folks who are brain damaged, who have intellectual impairments, yes, they have seizure disorders, but it is not that kind of. The fact that they have seizure disorders does not mean anything like that. So please don't misconstrue it. You're in school, I'm sure, with both faculty and students and staff members who unfortunately have seizure disorders and have to take medicine for them. It's not terrible. There's so many factors. I won't even get into it because I'm not qualified to speak on it. But see, 1% of the population is a lot. Figure if there's 4,000 students here, what does that tell you? 40 of them probably have seizure disorders. That's 1%. So all those, I mean, if there's 6,000 people on campus and there's 60, I mean, and that's just, you have to always look at the numbers for these types of things. There's lots of things, and mostly, fortunately, they do get an aura or a sense, oh, something's coming on, and they can lay down somewhere, try to get something around their head. If they're fortunate, they might have something like they have a padded tongue blade or something they can put in their mouth that they might carry in their purse or in their bag or have handy in their pocket. Okay. In children, it's like they disappear. They call it petit mal. I'll be talking to you like this, and all of a sudden, and then they'll pick it right up again within a matter of seconds. And it's again disconcerting. The grand mal, now we call tonic clonic. These we call absent sutures. This is all the problems. I mean, there can be damage. The swallowing your tongue thing is not typically something that happens. That's really not something that's true. So there's lots of ways to treat it. I don't know enough about them, wouldn't tell you. I'm not getting into consciousness or judgment and blah, blah, blah. Another word for fainting is syncope. And again, this is the distinction. Inadequate blood flow, emotional stress. People faint for different reasons. Overheated. I mean, it's just, I've seen all sorts of those things. Uh, my daughter faints when somebody has to stick a needle under her. She faints. You're just getting a regular blood test. She'll faint. We know it. Make sure she's sitting down or possibly lying down, and it's fine. Then she gets up and she's fine. So this is different. It is not the same as sleep. Your body is shut. Let's say it's in a recovery mode, and hopefully it will recover. It's almost like repairing itself internally. So you're basically not using things the same as, and then the idea, the ethical definitions, etc. I'm not getting into sleep and rapid eye movements and all that good stuff. Anything like that. I'm going to stop it very soon. Yeah, I'm not going to get into narcolepsy and cataplexy or insomnia. Okay. We'll make this the last thing. The meningeal layers. The meninges are the coverings of your brain. And yes, when they're inflamed, we have meningitis. And you'll learn about that in many of you basically. And you'll find out that viral is a little bit more treatable than bacterial, which is a life-threatening emergency. They are the covering protective layers of the central nervous system, including the brain and the spinal cord. The blood vessels are within them. The sinuses are within them. Those are areas where the blood flows and sinuses are like flat, wide veins. So all the blood in there is enclosed in there. As it's returned to the general circulation, goes back to the heart, protects it. Okay, it forms. You didn't see them because most of the time Dr. Robbins doesn't have the ones with the dura with the meningeal layers on them. The cerebrospinal fluid is in there, and it creates the divisions in the skull. I mean, it has thickened areas that separate, let's say, the right side from the left side, and the juncture between the cerebellum and the cerebral area. Big names for you. 3 layers. Dura, hard, durable, matter, material. Pia, soft, delicate, directly on the brain and the spinal cord. It's adherent to it. You can't really see it. It's just there. You have to identify it microscopically to know it was there. The dura is outside, strong, thick, tough. You can play tug of war with it. And arachnoid matter is delicate, and it gets its name because arachnids are spiders. When it looks like little strands when the anatomist looked at it and looked like a spider web. But in that space between the arachnoid and the pia, that's where the cerebrospinal fluid is. Don't forget that. It's in 2 areas. It's in all those ventricles in the central canal, that basically internal fluid. Lateral ventricles, third ventricles, aqueduct, central canal, blah, 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 blah. And the subarachnoid space. That's where the cerebrospinal fluid is located. Good diagram. These are called arachnoid. I talked about overflow valves. Remember, you're constantly producing the cerebrospinal fluid. Well, if you're producing basically a pint a day, you have to siphon off a pint a day because if you have too much, it's going to cause problems, or not enough, it's going to cause problems. And they feed into these what are called sinuses. Sinuses are located. The dura in the brain is unique because it has a thick area that adheres to the cranial bones. It has an area that adheres to the brain itself, and it's split. These little divides are called sinuses, and that's where the cerebrospinal is found. I beg your pardon. That's where the cerebrospinal fluid gets siphoned off, so it does not effectively it's an overflow valve, so it does not overload the system in here in the subarachnoid layer, and basically gets pulled into these sinuses. So you have the periosteal layer adherent to the brain, split in certain areas, not all, but many, and the meningeal layer directly on. And you can say meninges or meninges, doesn't matter. Here's the arachnoid. It really isn't that well defined. There's a thin coat, but you can see really it's defined. The fluid is within here, as well as you can see there are blood vessels in here. Okay. but they're not in quote unquote in direct contact with the fluid. Just that's what happens when we talk about a bleed. A subdural bleed would be trauma, and a bleed there we would call a subdural hematoma or a subarachnoid hematoma in these areas, and they're typically from trauma. Somebody hitting their head, somebody getting into a big fight, car accident, pick a reason. You have to be very careful with those, and they have to, and effectively, you have to keep a watchful eye called a neuro watch. You'll learn all about that to make sure one of these is unformed, because if it is, you effectively have to drill a hole in their head and siphon off that fluid, because if not, they're going to die. Very simple. And the pia is directly adherent to it like that. So it's very unique. And these thickenings of the dura mater, the meningeal layer, are called falx. I don't know whether you talked about those in anatomy, FALX. And they're the dividers, like for dividing the right side and the left side. There'll be another big falx dividing the cerebrum from the cerebellum, etc., etc. And they have a lot of thickenings that are in them. And it's sort of within these thickenings that we find these venous sinuses. And those sinuses eventually will flow into and part of the jugular vein, probably the internal jugular vein, and eventually that leads and becomes what's called the superior vena cava. So, two layers, fibers connect this tissue. Big, like I said, I'm not kidding about the tug of war. I used to have human dura when I had an advanced a graduate anatomy course at Chatham, and it was you have to slice through it. You can't just pull it apart. Periosteal layer to the surface of the skull, brain, not spinal cord, very important. We're going to get to that next time, next lecture. Meningeal layer on the brain itself, extends all the way into the vertebral canal, has spinal dura. So, I mean, that's a big distinction. Don't forget that. And the layers are mostly together, but when they open, there are these venous sinuses that collect venous blood from the brain and empty into the jugular vein. It's important. Just septas and divide. These are the septa, and they have fancy names you don't have to know. That's anatomy, kind of what it looks like. These are where these sinuses are. Don't have to go crazy about that or that. The arachnoid, the spider web look. So there's a little space where, unfortunately, this is where bleeds occur in the subdural. You hear about subdural hematoma, but the subarachnoid space, cerebrospinal fluid, and the big blood vessels. And the arachnoid granulations, again, permit reabsorption. I like the idea of an overflow valve. And the pia, delicate and covering the brain. And when it's inflamed or infected, then we have encephalitis, and it can be life-threatening. And we'll get to why we have to do a lumbar puncture or spinal tap to make the diagnosis. We talk about it both in micro, and I'll touch on it a little bit here in my next presentation. Cerebrospinal fluid, just in general, again, it's the cushion, buoyancy, so it's flow. It's not because if the brain by itself, gravity alone would push it into the foramen magnum. And so all these things, and we're remarkably resistant. Watery solution from the plasma, less protein and different ion concentrations. So it's I like to think it's like a filtrate or an extract of plasma. It's like plasma, but different. And again, those choroid plexes, and with the aid of the ependymal cells, they kind of finalize this interesting fluid. Here's the plexus, capillaries, enclosed by pia and ependymal cells. They have a role to play with circulation, ion pumps, which control the composition. That's what I mean. They're finalizing it, cleansing it, cilia beating for it. And again, if you do the math, that's about 450. It's not quite a half a liter, and that's almost exactly a pint that we make every day. And we so effectively replace it. And they're showing you the flow. If you look through all of this, oops, and you can see it's produced in these plexes in the lateral, third, fourth ventricles. The exit points are here. Three of them are here, medium, and one on either lateral side I talked about before. And when they go down the central canal, they can go all the way to the bottom and flow out. And there, this is all in the subarachnoid space, which you're looking here in the exterior. Okay. And the interior is in all the aforementioned ventricles. Let me just show that. And this is what happens if it blocks that fluid circulation. significant damage. In newborns, basically the head enlarges, and you have to address that typically with a shunt. In adults, this is the problem. I mean, it's life-threatening, so you have to find a way to drain it, whether you have to put a shunt in an adult or just basically open a hole and let the fluid come out and hope it heals. There's hydrocephalus. And this is the blood-brain barrier. And see, must pass through the lining of the epithelium because they can't, remember, they're continuous, but those tight junctions means that water-based substances can't get through. Okay, not through or around the endothelial cells. It's watertight, and the astrocytes surround that as well, and that basically is the blood-brain barrier. Simple diffusion, lipid-soluble substance, blood gases, back and forth. Those are the types of things, and then certain amount of facilitated diffusion because glucose is the only thing your brain needs. You need specific ions or specific amino acids, say for making neurotransmitters or other protein-like substances that are there. So it moves substances important to the brain, such as that. Okay. And they're showing you the difference between a typical capillary with these clefts, and you can see the clefts are not present, and the astrocytes being surrounding it and reinforcing it, as we talked about before. And you see, and even the additional things in the basement membrane, and this is why it's weaker there. So the vomiting center, the hypothalamus, monitoring compensation chemicals of the blood, temperature of the blood, etc., etc. Things that pass quickly, that are non-polar covalent and very compatible with passing. For instance, drugs that are part of that. So we have non-polar covalent things, oxygen, carbon dioxide, those kinds of things. Alcohol passes through. A lot of medications that are engineered, for instance, pain pills, narcotic analgesics, they pass easily. And so we are very cognizant of the blood-brain barrier. Concussions are temporary. Contusions imply permanent damage. And we're always concerned about swelling and things. So when someone has a concussion, the implication is it's temporary. But if you get more than one concussion in rapid succession, it can be a very, very long recovery period. Even if there may not be permanent damage, it can really affect you in a lot of different ways. So concussions that occur with some degree of frequency can lead to permanent damage. Contusion is direct permanent damage that's there. And when you don't have blood supply, it's a stroke. And what happens because of that, and I was commenting on it before, when ischemia means lack of blood supply. So death from lack of blood supply is what a stroke is, or a cerebrovascular accident. And what happens is the nerves now produce a lot of the glutamate, which is consistently excitatory. And it worsens the condition, and it's what causes those spasms. Obviously, all the muscles, basically, the only information they get is fire, 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 because that's the chemical, the exciting chemical, glutamate, was produced by that passageway, and that's when you get the hemiplegia and things like that. These are not mini strokes. I yelled about it before. Okay. And we'll talk about, and you should hear about TPA with stuff that they can use to treat strokes. But then we talk about that in the blood unit, or whoever does it. If you're not clear on it, just give me a holler. And so a TIA or TIE, we usually attack or episode, they used to call mini strokes, but they're not. It's a warning sign. It's an alarm clock. For some reason, brain might, people act differently. They see double. They might black out. You've got to find a reason for that. All of a sudden, they don't know where they were. And you have to understand that's what's called a TIA. And I don't want to get too much into Alzheimer's. And maybe I'll save that for another time. I don't want to give it short shift. All right. This should do for now. It's a pretty good complete lecture for me not being available on this Friday to be in. And thank you very much. Have a great break if you're listening into it before we return.